Glycine is the smallest of the 20 amino acids commonly found in proteins, and indeed is the smallest possible. The formula is NH2CH2COOH. Its codons are GGU, GGC, GGA, GGG of the genetic code. Glycine is a colorless, sweet-tasting crystalline solid. It is unique among the proteinogenic amino acids in that it is a chiral. It can fit into hydrophilic or hydrophobic environments, due to its minimal side chain of only one hydrogen atom. History and etymology. Glycine was first isolated from gelatin in 1820. The name comes from the ancient Greek word gamma lambda upsilon kappa upsilon sigma, sweet-tasting. Production. Glycine was discovered in 1820 by Henri Braconnot, who boiled a gelatinous object with sulfuric acid. Glycine is manufactured industrially by treating chloroacetic acid with ammonia, ClCH2COOH plus 2NH3H2NCH2COOH plus NH4Cl. About 15 million kilograms are produced annually in this way. In the USA and in Japan, glycine is produced via the Strecker amino acid synthesis. There are two producers of glycine in the United States. Chatham Chemicals, Inc., a subsidiary of Mumbai-based Sun Pharmaceutical, and Specialty Chemicals, Inc., which purchased the glycine and naphthalene sulfonate production facilities of Hampshire Chemical Corp., a subsidiary of Dow Chemical. Chatham's manufacturing process occurs in batches and results in a finished products with some residual chloride but no sulfate, while GEO's manufacturing process is considered a semi-batch process and results in a finished products with some residual sulfate but no chloride. Glycine is also co-generated as an impurity in the synthesis of EDTA, arising from reactions of the ammonia co-product, acid-base properties and structures. In aqueous solution, glycine itself is amphoteric. At low pH the molecule can be protonated with a pKa of about 2.4 and at high pH it loses a proton with a pKa of about 9.6. The nature of glycine in aqueous solution has been investigated by theoretical methods. In solution, the ratio of concentrations of the two isomers is independent of both the analytical concentration and of pH. This ratio is simply the equilibrium constant for isomerization. Both isomers of glycine have been observed by microwave spectroscopy in the gas phase. The solid-state structure has been analyzed in detail. Metabolism. Biosynthesis. Glycine is not essential to the human diet, as it is biosynthesized in the body from the amino acid serine, which is in turn derived from 3-phosphoglycerate. But the metabolic capacity for glycine biosynthesis does not satisfy the need for collagen synthesis. In most organisms, the enzyme serine hydroxymethyltransferase catalyses this transformation via the cofactor pyridoxal phosphate. Serine plus tetrahydrofolate glycine plus N5, N10-methylene tetrahydrofolate plus H2O in the liver of vertebrates. Glycine synthesis is catalyzed by glycine synthase. This conversion is readily reversible. CO2 plus NH4 plus N5, N10-methylene tetrahydrofolate plus NADH plus H plus glycine plus tetrahydrofolate plus NAD plus glycine is coded by codons GGU, GGC, GGA and GGG. Most proteins incorporate only small quantities of glycine. A notable exception is collagen, which contains about 35% glycine. Degradation. Glycine is degraded via 3 pathways. The predominant pathway in animals and plants involves the glycine cleavage enzyme. Glycine plus tetrahydrofolate plus NAD plus CO2 plus NH4 plus N5, N10-methylene tetrahydrofolate plus NADH plus H plus. In the second pathway, glycine is degraded in 2 steps. The first step is the reverse of glycine biosynthesis from serine with serine hydroxymethyl transferase. Serine is then converted to pyruvate by serine dehydratase. In the third pathway of glycine degradation, glycine is converted to glyoxylate by D-amino acid oxidase. Glyoxylate is then oxidized by hepatic lactate dehydrogenase to oxalate in a NAD+ dependent reaction. The half-life of glycine and its elimination from the body varies significantly based on dose. In one study, the half-life was between 0.5 and 4.0 hours. Physiological function. The principal function of glycine is as a precursor to proteins, such as its periodically repeated role in the formation of the collagen helix in conjunction with hydroxyproline. It is also a building block to numerous natural products. As a biosynthetic intermediate in higher eukaryotes, delta-aminovalinic acid, the key precursor to porphyrins, is biosynthesized from glycine and succinyl-CoA by the enzyme ALA synthase. Glycine provides the central C2N subunit of all purines. As a neurotransmitter, glycine is an inhibitory neurotransmitter in the central nervous system, especially in the spinal cord, brainstem, and retina. When glycine receptors are activated, chloride enters the neuron via ionotropic receptors, causing an inhibitory postsynaptic potential. Strychnine is a strong antagonist at ionotropic glycine receptors, whereas bicuculin is a weak one. Glycine is a required coagonist along with glutamate for NMDA receptors. The LD50 of glycine is 7,930 mg per kilogram in rats, and it usually causes death by hyperexcitability. A 2014 review on sleep aids noted that glycine can improve sleep quality, citing a study in which 3 grams of glycine before bedtime improved sleep quality in humans. Glycine has also been positively tested as an add-on treatment for schizophrenia. Uses. In the U.S., glycine is typically sold in 2 grades: United States Pharmacopeia, and technical grade. Most glycine is manufactured as USP-grade material for diverse uses. USP-grade sales account for approximately 80 to 85% of the U.S. market for glycine. Pharmaceutical-grade glycine is produced for some pharmaceutical applications, such as intravenous injections, where the customer's purity requirements often exceed the minimum required under the USP grade designation. Pharmaceutical-grade glycine is often produced to proprietary specifications and is typically sold at a premium over USP-grade glycine. Technical-grade glycine, which may or may not meet USP-grade standards, is sold for use in industrial applications, e.g., as an agent in metal complexing and finishing. Technical-grade glycine is typically sold at a discount to USP-grade glycine. Animal and human foods. Other markets for USP-grade glycine include its use as an additive in pet food and animal feed. For humans, glycine is sold as a sweetener, taste enhancer. Certain food supplements and protein drinks contain glycine. Certain drug formulations include glycine to improve gastric absorption of the drug. Cosmetics and miscellaneous applications. Glycine serves as a buffering agent in antacids, analgesics, antiperspirants, cosmetics, and toiletries. Many miscellaneous products use glycine or its derivatives, such as the production of rubber sponge products, fertilizers, metal complexants, chemical feedstock. Glycine is an intermediate in the synthesis of a variety of chemical products. It is used in the manufacture of the herbicide glyphosate. Research and development. Glycine is a significant component of some solutions used in the SDS-PAGE method of protein analysis. It serves as a buffering agent, maintaining pH and preventing sample damage during electrophoresis. Glycine is also used to remove protein labeling antibodies from Western blot membranes to enable the probing of numerous proteins of interest from SDS-PAGE gel. This allows more data to be drawn from the same specimen, increasing the reliability of the data, reducing the amount of sample processing and number of samples required. This process is known as stripping. Anti-aging properties. Glycine treatment may help reverse the age-associated defects in human fibroblast. Two genes that regulate mitochondria, GCAT and SHMT2, were found to affect age-associated mitochondrial defects. By changing the regulation of these genes, researchers were able to restore mitochondrial function or induce more defects in the fibroblast cell lines. In an interesting finding, the addition of glycine for 10 days to the culture medium of the 97-year-old fibroblast cell line restored its respiratory function. Presence in space. The detection of glycine in the interstellar medium has been debated. In 2008, the glycine-like molecule aminoacetonitrile was discovered in the large molecule HIMAT, a giant gas cloud near the galactic center in the constellation Sagittarius by the Max Planck Institute for Radioastronomy. In 2009, glycine sampled in 2004 from Comet WILD-2 by the NASA spacecraft Stardust was confirmed. This is the first discovery of glycine outside the Earth. However, glycine was identified in the Murchison meteorite in 1970. That mission's results bolstered the theory of panspermia, which claims that the building blocks of life are widespread throughout the universe.